we'll do a very quick review. Mitochondrial DNA is damaged just through the regular course of mitochondrial chemistry. It's very reactive chemistry. It's impossible to avoid the damage. As a result, there are massive deletions in this mitochondrial genome, which is over on the left. The mitochondrial genome codes for specific proteins in the mitochondria, and those are mainly to do with oxidative phosphorylation. Here you can see complex 1, complex 3, complex 4. When this happens, certain proteins are no longer produced, and when these proteins are no longer able to be produced, then oxidative phosphorylation cannot take place because it's missing key components. When this happens, there is a resulting shift in NADH and NAD+. NADH is the carrier molecule that carries electrons to the electron transport chain. And through oxidative phosphorylation, NAD+ is regenerated. It loses its electrons and hydrogen and becomes NAD+ again. But when oxidative phosphorylation is not taking place, NAD+ cannot be regenerated. It recycles the NAD+. Since NAD+ is not regenerated, in order to get more NAD+, because it's going to need it for the citric acid cycle and other chemical processes to keep the cell alive. So rather than die, what it does is it gets rid of these electrons through some other mechanism, which is by the plasma membrane redox system. The plasma membrane redox system, now that it has all of these extra electrons to deal with that it's normally not handling, it's trying to get rid of these all throughout the cell. But what ends up happening is that other passing molecules end up picking up these electrons and they become radicalized. And if they're stable enough, like low density lipoprotein, then they can carry that electron and oxidize something else in a different part of the body, thereby spreading damage to other parts of the organism. So that is the damage in a nutshell. Let us talk about now the two main proposals that are going to deal with this. One is called allotopic gene expression. The other one is protofection. And before we go into more detail about it, we have to go into a little bit of background about the mitochondria. But first of all, the mitochondria used to be its own independent organism. It had its own genome, and it still does to some extent, but it had a full complement of genes. Rather than having what is now 37 genes, it used to have several hundred. Some people estimate 4, 5, 6, 700 genes. Now all that remains are 37 genes that code for 13 proteins. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. 13 proteins. And this is because of exactly this problem. The DNA gets damaged in the mitochondria. The chemistry is very reactive in there. It's susceptible to a lot of damage. Whereas in the nucleus, where the nuclear DNA is, it is much safer there. It's shielded. So, what's been happening over time is that the mitochondrial DNA, the proteins that code for everything in the mitochondria, have been shifting over to the nuclear DNA. It's being housed in the nucleus now. So now how it works is if the mitochondria needs something, the proteins are coded for in the nucleus. The protein is then built in the cell body like everything else in the cell, and then it's sent to the mitochondria. And the basic idea of this allotopic gene expression is just to continue this, to move this remaining genome. That's a bad arrow. This remaining genome into the nucleus just to finish the job. Now, the only problem with that is it's not so simple. Otherwise, evolution would have already finished the job. There's one thing that all of these proteins have in common inside of the mitochondria. They're all very hydrophobic, which means is that they're repelled by water. Water scrunches it up into a little ball, which is fine because normally that's the shape it's supposed to be anyways. The problem is that the way it is shaped, it can't get into the mitochondria or out of the mitochondria. There is a gateway in the mitochondria that allows certain things to enter and certain things to exit, just like the way some things can enter a cell and some things can't. In the mitochondria, it has its own gateway. And this is called the Tim-Tom complex, and it's not a mental disorder. It stands for translocase, inner membrane, translocase, outer membrane. Tim-Tom. The difficulty with these proteins is that since they're bent up in these shapes that they're supposed to be anyways, they can't fit through the Tim-Tom complex. And think of it like this. If you're trying to move furniture into a house or out of a house, some of it is fine if it's got to fit through the doorway. Some of it's fine if it's through the doorway, but some things, they don't fit through the doorway. So what do you have to do? Assemble them inside your house. And that's basically what's going on in the mitochondria. These 13 proteins, they don't fit through the doorway, so they've got to be assembled inside the house. And while this seems like a big problem, this is what the whole field has been working on right now. So we'll go over some examples of what's been going on. So there are two main ways of dealing with this. One is to find examples in nature where evolution has already done the work for us, which has been quite a recent discovery. 99% of all animals have the same problem we do. They've got these 13 proteins, they don't fit, that's a problem. But recently there was a species of algae, when they're decoding its genome, and then decoding its mitochondrial genome as well, they noticed that 6 of the 13 proteins weren't there, so they were saying, what's going on? And it's not that they weren't there, they just were in a place they didn't expect. They were in the nucleus. With the mitochondria, they all perform the same function. They're all relatively close between species. And what has been done, and that's where I'm going to bring up some of the studies, this is the first example I could find of using the protein of a different species in a human cell. So what they did was they took this algae and they didn't do it for all the genes, they just took one. They did ATPase6. ATPase6, that is, there's here it is, ATPase6. So you gotta understand one more thing too about this study of aging. Since aging is not classified as a treatable illness by the FDA, you can't really conduct a clinical trial for something that is supposedly going to cure or be a relief to some age-related problem, because you won't be able to get approved for it because it's not seen as a disease. So what ends up happening is, for people who are interested in this kind of thing, is they end up studying diseases that are trying to solve the same problem. And one of those disease states are called mitochondriopathies. And basically what a mitochondriopathy is a genetic disorder, an inherited genetic disorder of the mitochondria. Some people have different genetic disorders where they're born without being able to produce certain proteins. Well, some of those can be related to the mitochondria. So they study diseases where different proteins are either not there or they're mutated. So if it seems bizarre that we're only looking at one protein at a time, or very few, that's the idea. Someone's looking at a disease that is mutated ATPase6, and what they're trying to do is use the gene therapy to replace that mutated gene. But here is an example where this idea of allotropic gene expression was successful was the first time it was, at least that I could find. This is in 2002. And what they did is they took a copy of that algae's ATPase6, which was able to pass through water and able to enter the Tim-Tom complex of this algae and see if it worked in human cells. And it did. It was relatively successful. It was not 100% success because, let me see. Here we go. The amount of mature polypeptide was 40% of the total expressed protein, implying that importation into human mitochondria was relatively inefficient. We believe that's similar to what we observed upon allotropic expression of human ATPase6. Unprocessed precursors present at relatively high levels in isolated mitochondrial fraction were either loosely attached to the mitochondrial outer membrane or were attached but not imported efficiently. And I read this whole article, and he, at least from my perspective, he seems to be of the opinion that it wasn't really an attachment problem, but more of an importing into the Tim-Tom complex. So while it was moderately successful, it was not 100% successful. But that's fine, because at least they were able to prove the concept that some of it was able to get through. And this is an old one, and we'll continue on to a newer study. Oh yeah, and here's somewhere I won't bother looking for it now, but it increased ATP production by 75%. So that means it is helping up somewhat the ATP production in these cells, but it was not restored to normal levels. But this is just the first one. Here is one that's done a year later, 2003. And actually it was done in Miami, which is pretty close by to where I live. I go down to Miami all the time. Actually, I saw Avicii there not too long ago. So maybe next time I go down to Miami, I'll see if these guys are still teaching over there or something. Maybe they'll give me an interview or something like that. And they'll probably be able to better explain what's going on as of today. I'm kind of looking back because I don't really know what's going on as of right now, but I'll try my best. So they were successful with one of the proteins, ATPase6. These guys tried 3 different ones. They tried ATPase8, apocytochrome B. Forgive me if I'm not pronouncing these right, because I've never heard of them in my life. I've only ever read stuff like this. I've never actually heard them. And then ND4. And in this study, ATPase6 was incorporated successfully. It was very successful, while the other 2 they tried were unsuccessful and in fact led to cell death. And it was the same problem. It just did not get into the Tim-Tom complex very well. But ATPase8 was. So here you can kind of go through. I'll put the links to all these on here. So here there's a whole list of all the different things they tried. They tried just regular, and then for the other 2 that they're having problems with, they tried adding hydrophilic groups onto it. So basically, you've got a molecule, which you can do, and this is common for other parts of the body, is add hydrophilic groups onto it, which basically are attracted to water, to try to stretch out that molecule in order to fit it through somewhere. And then once it gets in the mitochondria, it cuts the extra parts off and just puts it where it needs to be. But they were unsuccessful this time. But let me show something. So here, you can see what a big success ATPase8 was. Here, we've got the fluorescence, so you can see how well it got where it needed to be. Here you can see that it was basically spot on. But here, see the Cyto B one? So look, it's a disaster. And then the ND4 one, it's a disaster as well. It was not getting in. And look, so you can see with the pictures that all the rest, then they tried adding like hydrophilic groups onto it, and it just didn't work out as well. So they got, there's everything here. You can look and see everything yourself. But these are pages long. I don't want to spend all day going over it. Here, what they noticed with the main problem was that there were, with the 2 that didn't work, ND4 and apocytochrome B was that they're not only were they not getting into the mitochondria, but they were completely jamming up the Tim-Tom complex, and as a result, they were getting this aggregate of hydrophobic protein building up outside of the mitochondria. And it kind of reminded me a lot of Alzheimer's, the way that beta amyloid builds up outside of the brain, and then it kind of just chokes everything else off, because you've got all this stuff, then it's just like all this like branches of stuff building up all over the place, and just being a nuisance. And the same thing is happening to this mitochondria. Since it's not getting in there properly, it's causing a dysfunction of the whole cell. It's basically having that kind of beta amyloid effect, but inside of the cell, and it causes cell death. So that's not something we want to cause at all. Yeah, but ND or ATPase A was a success, but also they were. I was trying to find out where. I couldn't remember where they were talking about ND4, but they were pretty disappointed with the other two, saying that there could be some of these proteins that are so hydrophobic that it may be impossible to do exactly this. Try to get a nuclear encoded version of it. But years later, I found this. This is from 2008. And this is a very interesting one. This was done by some French scientists, I think. Yeah, those are all very French names. Yeah, this is the French guys. But what they did, they took ND4, the one that the other guys a few years ago thought might be one of those proteins. They may not be able to get inside of the mitochondria. What this does is they took that ND4 gene and they put it into mitochondria, and they did it successfully, and it was extremely successful. Let me tell you how successful it was. And not only, I forgot to explain, these other two studies were done just in cell cultures. They weren't done on animals. This third one in 2008 was done on animals. It was done on rats. They took the ND4 gene, and ND4, the deficiency in ND4, there's a disease called Lepers Optic Neuropathy or something like that. It's one of the most common mitochondrial mutations where people have been wanting to do this gene therapy. But basically what they did is they gave these rats this mutation where they couldn't produce ND4, and what this one does, I think it's Lepers or something like that. It's probably on here somewhere. Lepers Optic Leber hereditary optic neuropathy. Basically what it causes is blindness. So they gave it to these rats, this mutated ND4 gene. Gave it to two sets of rats. They all got blind. Okay. Next what they did was they took this modified ND4 nuclear encoded gene, put it into the rats, and they found that the rats that they gave the gene to, they stopped having blindness, and the other ones went blind that didn't get it. So it was a proof that it was a success. And I liked this one because in this other one over here, these guys were pretty sure that they weren't going to be able to do it, but it just shows that over time it's amazing what people can come up with. Also, I thought it was bizarre here. It's not really bizarre, but see, look at this. The way they describe, I guess when they killed the rats, animals were sacrificed. I mean, it's not really that funny, but I've just never seen it written that way. I thought it was kind of, I don't know, it was just I just never seen it. And they keep going on to it. I in my head when I'm reading this, the whole time I'm thinking of a bunch of druids around a fire, in a lab somewhere. I don't know, but anyways, not funny, but it was kind of just alright. When you keep reading it, you can't help but just be a little bit amused. But yeah. But anyways, that's kind of where allotropic gene expression is. They're gradually getting, and these guys, in fact, at the end of this, they said they were going to try it in higher animals. So that's a little bit closer to having something for humans. And this is for Lepers' optic neuropathy, which is not aging related, but you have to understand any solution to a genetic disorder like Lepers' optic neuropathy, anything that's going to cure something like that will have the same kind of effect for what people who are trying to find something for aging are trying to do. They're both trying to do the same things for different reasons. So that's that allotropic gene expression, and basically what they've got to do is just find it for all 13. So they've been successful with a few, and there are some like cyto whatever it be that I don't think they've been successful with that one yet. But I give it time. There are only very few people working on this on the entire planet. You can probably count them on your hands. The next one, which is new, is called protofection. And protofection is different from allotropic gene expression. Protofection is basically transferring a new set of mitochondrial DNA into the mitochondria. Basically just replacing the old mitochondrial DNA with new mitochondrial DNA. And this is very, very new, so I wasn't able to find so much on it, but I found this. Mitochondrial gene therapy arguments, physiological in Parkinson's disease. So they're modeling this after Parkinson's disease, and this is... So let's have a look at what their deal is. Oh yeah, this finding suggests that Parkinson's disease pathogenesis, at least in its later stages, includes clonal expansion of uniquely deleted mitochondrial DNAs within aging neurons, leading to loss of bioenergetic capacity. Such neurons would be impaired in normal synaptic function and vulnerable to death. So with a lot of these high energy cells, brain cells, eye cells, muscle cells, heart cells, anything like that, they burn a lot of energy. So a lot of the other diseases that people are studying are kind of like long-term effects. You probably know is that brain cells and heart cells are not replaced when they die. So a lot of pathologies where we've got these mitochondrial damages, that is very destructive for certain diseases like this. So someone who's got a problem with their mitochondria, it's going to facilitate that. That's why people go blind with lepers, because they're not producing as much ATP. The cells start dying off because they need that energy. While some other cells, they won't die because they don't need as much energy. But these cells, I can't remember what the statistics are on your brain, but it uses I can't even remember. It's like 30% of all of your energy, and if you're thinking or something like that, like I know when I play chess sometimes, especially if you play a game like blindfold chess, it's just exhausting. You'll be physically worn out from playing, just from thinking so much. But anyways, they have the same kind of problem that we talked about in the first video, about how those mutated mitochondria proliferate because they're not being selected against by the lysosome, because they're not undergoing oxidative phosphorylation. So they're not being selected against. So if they're able to restore this oxidative phosphorylation, then these cells will be okay, and then at the same time we won't have all of this damage occurring because they're exporting garbage out all over the place. But anyways, so have a look here. There's a picture. So basically what they're doing is they're making a molecule that is going to go into the mitochondria and replace the DNA. So now we have new DNA that's not messed up, and it's going to code for regular proteins. And what was found, and which I found really astonishing, was that there was a 20 to 30 fold increase in gene expression. So what they did is they took messed up mitochondria genes that weren't undergoing oxidative phosphorylation, they treated it with this stuff, and some of them were successful and some of them weren't, but some of them were incredibly successful. And one culture on here, it's called PD59. After 10 weeks of this treatment, in the beginning, it was completely anaerobic. There's no oxidative phosphorylation at all. Let me see if I can find that on here so you can look at it yourself. P59. The most dramatic effect of treatment on the intact cell respiration was in PD59, which was functionally anaerobic at baseline with low mitochondrial gene copy numbers. 10 weeks after treatment, the respiration rate was restored to normal. So that's pretty astonishing right there. But that's just the basic idea. There are other studies out there, but I mean, I could spend all day going over this. I just wanted to give you just a very, very bare bones understanding of what it is people are doing out there as far as age-related stuff, and maybe you're interested in Parkinson's disease or these other mitochondriopathies, but that's basically where it stands today. And they seem, as you can see, they seem pretty promising. And probably if they did get a good amount of money and there were actually a good amount of people working on it, they could hypothetically figure it out rather quickly, because just with very few people working on it, you can see the progression of these studies. 40 is like basically very limited help, and then here you already had one of them very effective, and then not too long after that, one of those that was completely toxic in the cell, they actually managed to make it work in a living animal and prevent that animal from going blind. And then you have this new one over here, which is really, really recent. I think after I stopped reading this years ago, I think it was just brand new, and I just read it again recently, and I'm just really astonished at how quickly these things have progressed. So there it is for you. If I find out anything more about these, like I said, next time I go to Miami, I might see if I can stop by that school. Maybe they'll talk to me or not. Maybe they will. I don't know if they will or not. But if I do, maybe I'll record it and I'll put it up here. That way you'll get some more information about it. But there it is. I'll move on to the other types of damage later on. But this should about sum it up for age-related issues related to mitochondria.